sidebar conversations. But really what they're doing is that working with either internally their own research programs and applying things forward to use this, or they're working with the educators as far as, okay, we've got this research protocol. Here's how we're going to get this out in the field. And then that way it all kind of dovetails. So we don't have things going everywhere. Beyond that, though, we do have folks like Brian and Hans, and to a lesser extent, Joe, is that they're going to be around too to make up those one-off type things to where, okay, we just want to see if this one idea works and then maybe expand upon it from there. So they're responsible for the trainings for the staff throughout the year, and then also the overall research and surveying that the extension staff are doing throughout the state. So what this kind of looks like, this map changes all the time, even though really, especially from the last 3 years. So this is, it was an idea 3 years ago, 2 years ago was a beta, and last year was the first full year that we kind of rolled it out. Give you some idea as far as distribution. And so with that, it works, if you will. And we also have several people that did the field survey. So we have people that are involved, but then we have the field survey. Let me go forward just a little bit. What this kind of means to you and me is that this particular slide here is that we're going out, and our surveys are a little different. And so when we talk about survey, we're not going out and talking to the plants, of course, but we're going out 3 times in a given year, and actually that usually means about 5 contacts with the farmer, because then before we go out in the field, we're talking to the farmer as far as, okay, what do you want to look at? But then beyond that, what type of seeding rate you're doing, what is your pesticide program as far as regard weed control, insect control, fungal control. And that's done on the early side. And then we're out in the field, usually after emergence. Well, it's going to have to be after emergence, because that first go around, we're doing soil tests. We're also looking at emergence rates, any type of seedling diseases, things that are going on, insect pressure, weed pressures, how effective was the burn down control. In the middle of the year, the second time out, they're going out, and that's essentially at that period of time when you're at full bloom, roughly R1, R2 for both soybeans and corn. And so for those of you that are not familiar with that, that would be the time of year that you see the beans probably around between mid-shin to knee-high. And then you're going to see the corn is actually starting to silk. So that's going to, throughout the state, that's going to vary about 3 weeks in length, if you will. But it gives us that snapshot. And what we're looking at that time is, okay, so how is the plant doing? We're taking foliar leaf samples at that point in time. We did the soil testing on the first go around. Second go around, we're taking foliar leaf samples to see, okay, and we're doing it at a point of the plant that gives us the most information. So it's almost like junk in, junk out, which you're probably all familiar with, is that the idea of sampling the leaves that matter the most. And so we're not doing, on soybeans, we're not doing the old stuff at the bottom of the plant, and we're not doing the very tippy top new leaves. It's just below that, basically the oldest, most mature, or excuse me, oldest, least mature leaf. And so it's a fully developed leaflet or trifoliate for soybeans. And for corn, it's going to be the leaf that matters the most, which is the one on the ear shank, or the one that's attached to the cob if you're out there walking in the field. And so we send those in, and we get information with regard to their overall health and wellness. When we do the soil sampling earlier on, we're doing stratified, and we're also doing an average. And so in many cases in a farm setting, they'll either group them by soil type and test in that area, or they'll do what they call grid sampling, where systematically, if you just put boxes out there on a field, and so at this checkpoint, we're pulling a core, so on and so forth, and do an aggregated sample that way. We're going out in the areas that we're actually scouting, walking a W, if you will, and taking samples as we go. And then that is one sample that's blended together to represent that. And we do a 0 to 4 and a 4 to 8. So that allows us a couple of things. The 0 to 4 and 4 to 8 differentiation can indicate whether or not we have stratification. If you look at them also too, you can get some ideas as far as the overall health of the field. And then if it's in a soybean field, we take that 4 to 8 inch sample, we split that in two, and half of that goes to the soil nematology lab out in Missouri. And so they have a soil nematology lab out there in the Boot Hill of Missouri. It's been there for years, and that's kind of their thing, to where they're big on soil soil nematode testing. It's ran by the University of Missouri and their extension staff out there, but they directly cooperate with Darcy Talenko, who is our plant pathologist up at the university. And so they do the actual in-lab testing, if you will, and give the results back to Darcy. And so that's what happens with soil tests. With the leaf samples, when we're going out mid-season, then of course we're looking overall fertility, and that information goes to ANR labs, and then they give us up there in Fort Wayne, and they're the ones that then give us the soil fertility for both the soil and the foliar leaf samples that we send off. So this gives you an idea of what we're kind of looking at. And like I said, our survey is not what probably most people do. It takes quite a bit amount of time, and maybe in and of themselves, for instance, what I tell like farmers that I've talked to about when we do the soil sample, it really makes no sense to them, because when you're covering, you don't have one soil sample represent 30 acres, okay? But what it does for us at the state level is that it shows overall averages as far how the overall health, of the soils are in the state, and then by field within a region. And then over time, of course, you track that. So as far as a farmer's direct worth, it gives them some idea as far as, okay, well, on average, this is how my field's doing. They'll usually want a higher level of specificity than that. But then there's other things that we do that do have direct relevance to actually what they're doing out there. And then some of the internal conversations that we have amongst ourselves is that fine line, because it really does depend upon the producer, just like it depends upon any type of client that we're working with, or colleague, friend, neighbor, is that really how much information does that person want? And so every time we go out in the field, we do the survey, that data is collected, and then that educator has a print off that then they can then take to the farmer directly. But there's also times where I've literally called the producer in the field, and that's happened last year. It's happened in following years, or years previous, excuse me, to where I said, hey, you've got a problem, and there might be something you want to investigate right now. And so last year it was an issue with a weed out in the field that I knew that if he went along or hadn't looked at the field recently, that it was going to be an issue for them. And then likewise, if we're out there in the fields and we start seeing it's a dry year, which wasn't last year, it was in June, but not at the time where it would have been a problem with mites coming into the fields, where, hey, you might want to be looking at your border fields and consider possibly treating the borders before it gets into the field itself, that type of thing. So during the growing season, pre-plant, where I talked about early and mid, and then of course late season, we're doing some ideas as far as overall yield and health, and then disease pressure, things like that. The educators are strongly encouraged, and actually it's already through funding that we have through Indiana Soybean Alliance and Indiana Corn Marketing Council, Corn Growers Association, is that that barrier of us getting stuff to campus to then be evaluated is removed. And so we have them take samples not only for the overall fertility of the leaves, but then also send off samples, suspect samples, to the plant pest diagnostic lab. And if they have the time too, we also ask them to go ahead and send them samples, even if they know what it is, but they want to go ahead and confirm it. Because what happens in the background is that, and this would be the same thing with horticulture, forestry, or anything, is that every year that a certain disease is found in a county, then you'll notice, if you ever looked at the plant pest diagnostic lab reporting forms, they ask what county it's in. And so that then all feeds, it doesn't necessarily all the data as far as, okay, this person, this location, lat long, we're going to come and get you type thing. But it's just it's an acknowledgement, is that every time, okay, we confirmed a a sample of gray leaf spot or northern corn leaf blight on this date with this sample in this county. So once again, the farmer or the producer or the gardener for that matter just wants to know what problem they have. But what's benefit for the pathologists, both Purdue, Darcy Tillnico, and others, but then also nationally is, okay, that overall spread or plume, or what are the climate factors that are interrelating with all this, that's happening in the background over time when we develop these histories. So that's happening as well. And so, yes, we send off a lot of samples now, and it's all been funded. And so the educator doesn't have to worry about stressing the county budget, because I don't know if you've ever tried to ship anything recently, but in my opinion, shipping costs have doubled, if not tripled in many cases. And so I think the lab still only charges $11 statewide for the preliminary documentation of plant pest diagnostic lab up on the Purdue campus. It's only like $11 processing fee. If they do auger work, it gets a little more expensive, but still it's very reasonable. But the very sample that I will spend $11 on to take, it'll cost me $75 just to ship it to campus from Shelby County, which is only about 100 miles away, plus or minus a mile or two. So I mentioned this, I think I mentioned everything on this slide except for one thing too, is that with Mark Carter being on the team and a lot of us flying drones throughout the state. So I'm a licensed drone pilot myself, FAA drone license, is that every farmer that works with us, we try to get them at least a beauty shot of their field. And what I mean by a beauty shot would be up there, take a shot of the field itself. Some of us also have the enhanced ability to actually do what they call a stitched image, to where we will fly the drone over the field, and it'll take, depending on the size of the field, it could take several hundred pictures. And then they use AI and they stitch it all together. So what that allows you to do is that you got the picture of you can go from just looking at the field like Google Earth all the way down to being able to see individual leaves on either a soybean or corn plant. Not so much detail where you're seeing bugs on the leaves, but you can definitely see the tassels of corn and things like that. And as far as level of granularity of the picture for that farmer to then look at for the field. And even the glamour shots can give you some good information, because the nice thing about drones, when we're using them constructively in this regard, is that just a simple picture up in the air can show you a lot of things. Because when I explain to people in the ag community, is that it's really no more than a camera that you're putting 400 feet in the air. And so instead of you having the corn 6 feet over your head, if you will, is that now you can look down upon that crop and you can see individual plants, or you can see the entire field with just a zoom in, zoom out. Or by doing an offset shot, you can see things like color changes, things like that would draw your attention to it in the first place. And especially when we do the stitched imagery, you can literally drop a pin and then walk out there to that spot with your phone. So you can pick up tile lines that are messing up, planter boxes that haven't done quite right, applicator knives that messed up. Not where our job is not to point out everyone's mistakes. That's not the point, but it's to help them see what's going on. And so in that regard, drones are tremendously good, because you're doing it mid-season. So as the plant's growing, you can go and see this stuff versus getting the image of sitting up in the combine and wondering what happened. You might learn even early enough to take corrective action later on in the season, or excuse me, earlier on in the season. So with all this data we collected, what does this all mean? Is that it means that when we did the soil samples this last year, is that they were a little bit short. And so you think, oh my gosh, what are we going to do? Well, if I go one step further and say, okay, so we got 64% that are under our potassium thresholds. We have, yeah, and then 23% for phosphorus, that doesn't sound good. And then pH is starting, 2/3 of the fields are less than pH of 6. which for corn's a grass, so it actually likes a little bit lower pH, but that's not going to bother too much. Beans is a different story. But then when we go out and we look at corn later on, actual samples later on, is that kind of drops off. And so sulfur still not doing as well. But you can see here on the potassium and phosphorus is that those aren't the same. So even though the soil tests were showing that it might be a little bit short, the corn plants were able to scavenge it. And so, and to be able to find what they need. And a very similar story for the soybeans, and that we had, instead of 2/3 of the field, we had just under half of the fields under pH of 6 at the time of sampling, which had been, and realized too that there's some other factors. It depends on when they applied the lime, if they did that type of stuff. But anyhow, when we did the snapshots and doing overall samples, looking at those county fields, is that again, we had phosphorus was okay, but then with the potassium, almost 3/4 of them might have been a little bit low on their potassium. But then when we look at the actual leaf samples and send in those samples, then suddenly instead of almost 3/4 on the potassium, we're only 5% or beneath our thresholds. So what that's telling us, might be a little drier when they sampled the first time. Then we had some rains when they came around. It could also just be simple genetics. So that's one thing that I've learned with the crops. And so every year when I'm working with the press, they, what's the yield going to be? How are things going to go? And I always say it don't matter till it's in the bin. And what I mean by that is that I never count the Indiana crop out until we know that it's done. So, and the last time that happened would have been in 2012 when we had the drought, and really the beans did fantastically that year because even with that drought year, the rains came late and the beans just sat there and they bloomed, and then they lost their bloom and they brought it back, and then cycle after cycle after cycle. And then when conditions favored, then it set out all the everything pollinated and pod set, and the yields were pretty good on the beans. So the corn, not so much. We had a lot of 0-acre fields in Shelby County in 2012 as far as overall yields. And in some cases, I probably add insult to injury. I think it was when the farmers told me where they're getting 8 bushels per acre, because that's, you still got to go in, you still got to harvest it, but it's not even paying for the diesel fuel on a per acre basis to actually go in and get all that. So, all right. So we looked at our top 3 weeds. Once again, this is just in the fields we're looking at, how things are going. And I don't think this should be much of a surprise for anyone. It certainly wasn't the farmers. As far as early season and mid-season, that's really the times we're really looking at weeds. By the time fall rolls around, yes, if you have a giant ragweed or if you have a water hemp issue, then that of course is an issue. But when you actually do something about it, is more of that early season and maybe later on with the mid-season, depending upon what's going on. But this gives us, once again, it's an idea of what's happening in these fields and what level of control for the different diseases are out there. And so this is really like year 1 was official, and we have the year prior when we kind of beta tested. We had information we can see year after year. And I can foresee that probably 15, 20 years from now, we might simply be talking about different weeds because of selective pressure, things that can happen. As far as insects are concerned, we had out there with the mid-season with the corn, first Japanese beetles and soybeans. We're coming out there, and then but still, once again, just not that much. We didn't have much insect pressure, and then, but that is information in of itself. And what I mean by that is by when we look at disease pressure, is that one thing that we knew when these plants, especially corn, was beginning to tassel and we were out there in the field, is that there really wasn't that much disease out there to deal with at that time, simply because it hadn't rained, it wasn't moist. We were on the verge of, we were in a drought and maybe going into a worse drought in June, end of June. And so the disease pressure was very low. They might have still sprayed, but that being said, the pressure simply wasn't out there. And so we were able to document that both by samples and pictures, and then And then that, of course, gets backfed to campus. As far as later on, we have a survey at the very end where we ask the farmers themselves. And some people look at this one, but this is what your neighbor did as far as fungicide in corn and soybeans. And insecticide, just not very much. It's with the genetics that we have now, by the time it rolls around later on in the season, it's kind of it's a non-issue. There's specialty cases where we're doing food grade stuff that might be non-treated products, but in the most time it simply is not an issue. And we've shown that for years to where insect pressure is not nearly as high as it was 20 years plus ago when I first started. So one thing between 23 to 24 rolling into that is and things that we did last year was that we all need to have a training and work amongst ourselves and kind of tease some stuff. And so this last year we started working on a biological study, which will be rolled out even more in 24. We of course got the drone footage you already mentioned. We did the statewide surveys, and then we reached out to other states, and we'll continue to do so as far as trying to make this program even better. In 23, a couple of quick takeaways that we got was that indicated that the fields are able to scavenge enough of the nutrients. So even though the soil samples might have showed otherwise, the foliar samples kind of countermanded it. And then of course, with several farms setting yields records, including in aggregate statewide last year, that kind of proved the point too. And then because of the season that we had, as far as a dry time right after planting season, and then clear up into right before tasseling or at time of tassel, is that disease pressure was pretty light. And then of course, then we did get some rain, and the grain, both the ears and the pods filled out, and had a pretty fantastic year for most farms last year. In 24, there's several things we're going to try to push the biological studies out more and more. And for those of you that are unfamiliar, that's exactly what it sounds like, is that what bug juice in a bottle. And when I mean bug juice, I'm talking about either fungal or bacterial type of application, different ways to apply it, of course, different products, but what actually works. And I think Sean Castile said, Dr. Castile said that there was like 400 different products on the market right now. He narrowed it down to 100. And then we pushed out, I think, 2. We said 2 got pushed out to the county level as far as working with county educators. There's also a program that we're going to be doing that is available for farmers if they so choose. Is that Dan Quinn's working with the University of Illinois, and it's actually an entire consortium involving Texas A&M and several other states, the I states, of course, as far as rates, corn planting rates and other things. And it's pretty fascinating because they're using artificial intelligence, and instead of having maybe 14 or 15 replicated strip trials and using the entire field length, is that they're using AI to actually put, to me, it makes a field look more like a low resolution television screen. So instead of having 15, 18, 20 replicated strips out there in the field, or 30 or so, you might have several hundred. And so they're using the technology of training the planter and then getting the data back from the combine to tease that out as far as the changes from the various rates and placements and other things. Pretty cool. And the thing is, does it actually work? And so that's the nice thing about us working with the county and then working with the state staff and the farmers, is that we can make that cycle happen a little faster. And it's just more data points, which it makes the data that we do get more secure as far as the likelihood of people getting the same outcome as we get. I know that Darcy. Scott? Yes.
Oh, there was a quick question because you're kind of on the crops topic. There was a question about, was there a difference between crops' ability to scavenge nutrients till or no till fields. If that was something that they're looking at with these studies.
I can tell you last year, especially that the planting season that we had last year was that, and I think Sean commented about his soybeans, but in the fields that I had is that, especially my one no-till field with cover crops, during that second walk, or was it the first? It was the first walk in the field. It was rough. Not going to deny. But by the time it got in the bin, there wasn't a difference. And as a matter of fact, that particular field was absolutely beautiful when it came to the corn crop that they were growing. So the short answer is yes, it was visible. And then we documented it up at the agronomy farm at Purdue too. And there were some, especially with no-till, there were some obvious differences as far as maybe at time of planting, and because it might have been a little damp as far as compaction, and then we went in that dry cycle, that some of that was exacerbated. And so you really saw the physical difference, but by the time it came into the actual yield, I think it pretty much caught up. And we've run into that issue in the past too, when we were looking at sulfur. And you'll see where agronomists are pretty cagey on this stuff, and for a valid reason, is that sometimes you can visibly see the difference in the plant. But when it comes to the actual yield, the plant somehow makes do. And so when we did some sulfur trials in my neck of the woods, is that we were able to later show, okay, yes, it's both a color change. Yes, we see it. And yes, it does reflect on yield, but that is simply not always the case. So as far as their ability to scavenge nutrients in till or no-till fields, just remember too, it also depends upon the time of year we have too. And there's always that balance of the two. If we have a wet planting season, it's cool and damp, then your no-till is going to have a hard time going. But we haven't, and that's where the whole cropping season comes into play. And when you think about it, when we had pollination time, when it really mattered, and yield from a yield perspective, is that it's actually turned off cool at about that time. And so in that way, it kind of saved us. So if we were still in full-blown drought and it was still going to be 90 degrees at night, 85 degrees at night, then I think we would have had a much different result. But in that case, we did start getting a little bit of rain. These hybrids that they're planting now are very good at scavenging water and utilizing as well as nutrients. And then beyond that too, is that the temperatures. And so the temperatures backed off a little bit. If we go back to 2012, which what worried me a little bit in June this year, but we've got the entire cropping season. It's almost like a basketball game, right? Where we look really good in the second quarter, but what really matters is when that buzzer rings in the fourth quarter. And so and the farmers, I've learned over time, they do their own self-compensation as far as making what works for them the best. So do we have it? Do we see the differences? Yes. We saw it on the farm, even up at the even up at Acre, up on campus. But in the end, state yields are still record breaking. Okay, was there any more questions? Because I think that's kind of where we're at. So how does this, I saw the people that do all the gardening and stuff, how does this really impact you? And really it comes down to this, it just makes our educators and it makes the people on campus better at what they do, long story short. And what I mean by that is that we're still using the rust is still rust. And so rust might be on your plant in your garden. And we still have rust on the crops too. And so the ability to tease out what a fungal issue is, or a lot of the same insects are problems in both gardens as well as in fields, so on and so forth, and to better recognize and faster recognize. And then when we get stumped, to be able to more quickly send that stuff to campus, and for campus to be able to diagnose it, it just makes it makes everyone better at what we do. And so It's something that we've done all along. This is, but Purdue on the Farm is probably a little more structure to it, and it's a little more intentional. In other words, instead of an educator in the county by fits and spurts learning something, is that we're pretty a little bit more consistent or more aggregated, if you will, as far as, okay, well, here's a set of skills that we think everybody should know and be able to do and be able to do it well. And so those that have been around for a long time, it's just sharpening that knife a little better, getting that winter dullness off. And then for those that have never done it, then it's a way for us to team up and make them that much better. And then the other thing too that I just think can't be emphasized enough is that the cooperators that we work with, the farm community, can see pretty quickly the infrastructure behind us that allows us to do what we do. And then, of course, we share it. So that's the thing that we do that I learned even back when we had the soybean aphid outbreaks in 2003, 2005 era, was that a lot of the stuff that we did made it so that way the commercial groups, the co-ops and the plant health agronomists throughout the state can more quickly and better utilize it. So that's kind of what we do. It's not, we don't always take the limelight, but we definitely help with the overall infrastructure and getting especially the new stuff out there, and then watching the old stuff and cataloging and see how we're consistently doing and sharing that with the public.
Compare that because I like how you talk about the ag side of it. So I work with the turf side of it. So just think of all you've been talking about, but much smaller. And a lot of times those snapshots are the golf courses, baseball fields, football fields. So we see different issues with that. But then with the home gardens, that's a better snapshot because just like the fields and soil types are all different from the north of the state to the south to central, you do your best to just get that snapshot so we can see what's going on. Because I think you had mentioned or had you mentioned the Purdue Pest and Diagnostic Lab? I can't remember.
Yeah. Yeah, well, I just keep calling it the PPDL, but yes, you're right, Jeff. It's the Purdue Plant Pest Diagnostic Lab. And so, and like I said, we have so much bleed over. So it's good when Jeff first sees dollar spot that he knows that, and then other people, so we can expect it. I mean, we've got models, I think Michigan State put together as far as monitoring some of that based upon growing degree days and that kind of thing. But same thing with, we had a thing that happened, it was probably 10 or 15 years ago, because it's been a while, but and those that garden a lot will know what I'm talking about. But essentially, the nationwide seed stock growers for tomatoes accidentally inoculated their tomatoes with late blight. So it came from the grower that makes all those little plugs that are then put out in the big box stores, the Walmarts, the Lowe's, the Home Depots that they sell to. They came pre-inoculated with late blight. Well, we have growers here in Indiana for red gold, your ketchup growers, and we had at the time tomato growers in Shelby County. So it was pretty important as late blight samples were sent in and checked that then Plant Pest Diagnostic Lab was making that information available so we can kind of see that disease spread throughout the state. Because instead of it coming later on, like it always does, as the name kind of implies, we have it. We started out the growing season that year with late blight, which is a slightly different story. And so that's one thing to where, again, we're using our skills. And in that specific case, it's crossing over both on the commercial and on the home horticulture front.
And that just brings in too, because we've mentioned the ag side of it, row crops, turf, but then even because as extension educators, we cover a lot more than that. So invasive species, whether it be a plant, whether it be an insect, I know the DNR works quite a bit with that, but if you have a question of where to take some, for instance, the spotted lanternflies in Huntington County, which is one county to my east, that's something you go to your educator for, and then we will work with the DNR, we'll work with our entomologists to, again, get that information out. And so I don't know if you've seen a difference, Scott, but I remember when Emerald Ash Borer came through, it took forever to permeate all this information. But then now we're just kind of getting a little quicker. Like with this ag on the farm, you're seeing the issues kind of within a year time span, and the cycle just gets a little, but I'm going to say tighter. Am I hitting
Yeah,
that okay?
well, but that's what we're supposed to do, right? That's, to me, that's would have been an internal expectation of us to better serve the public, to shorten that feedback loop. And it's all ran by humans. Humans are all involved. And so there's always room for improvement. But little things too, like you're talking about the spotted lanternfly, is that okay, we have that, but we have the tree of heaven that we've had here for decades. And so it was an invasive. And what's its favorite plant to grow on as far as spotted lanternfly? The tree of heaven. And so it's the ad education. It's never ending. I remember when the soybean aphids first hit Indiana. It hasn't been a thing for several years. So knock on wood, it doesn't become a thing again. And if it is, it's usually double crop beans. But there was a time it was in the original. It wasn't in the double crop. It was in the actual normally seeded bean season, if you will. And, people were calling every single white insect cast on the back side of a leaf a soybean aphid. And it wasn't necessarily that the case. So that's, yeah. So we try that. Now, see, we got a question here. How do neonicotinoid treated seeds plant affect any of the fungus invasive of the row crops? Those affect the native pollinators in the garden. See here. So I know that that is a very hot topic, if you will, as far as, and I know that Christian Krupke and them have worked on some of that, and even looking at the dust. The thing with the neonics, it comes down to handling and application, things like that. And I know that those that are closer to the source, if you will see that the handling procedures and the binding on those seeds are definitely more, it's a lot more stringent than it was within even the last 5 years, as far as that label's concerned and how those products are being handled. So yes, I mean, we know that bees do not like neonics at all. That being said, people use 7-Dust all the time. So
I think for that, I'll put in the chat. There is a resource called Drift Watch. I'm not sure if you're familiar with that, but we use that quite a bit. In terms of if you have a hive or some high value crop, you can use that link.
right. One
But
thing I will say about Drift Watch, Jeff, though, is that is for a commercial. And
oh, gotcha.
now bees are different. So bees are something where they will allow you. So, and I think it's called Bee Watch. So they've got Drift Watch. They subdivide it down. I'm not sure why, but they do what they do. But if you are commercially growing vegetables or commercially growing, let's say flowers for the market in Indianapolis or something like that, then you can also designate to that. And that's not something that people can just drop a pin like on Google Maps or something. You will actually get a phone call and verification to the state chemists. So where that comes into play is that it's somewhat helpful for farmers in that regard, and that if it's actually on that map, then it's something that should be adhered to or respected. And I'll leave it at that. Not saying that gardeners shouldn't be, or gardens shouldn't be respected, but there is a size and scale at some level that it does take to get on the Drift Watch website. And I am not that judge and jury.
Scott. Yeah, I just use it with my farmers because again, we teach a lot of farming stuff, so I make sure they know that that's out there too. So, any other questions from the audience? We still have some time. I'll go ahead and stop the recording for that. But Scott, if you're okay to hang around for a bit, otherwise,